demonstrating that the caterpillar Helicobacea grew larger on the tomato fruit tissue in comparison to the tomato leaf tissue. By the way, this chart was actually graciously donated by MR. However, as you can clearly see, the caterpillars that ate upon the tomato fruit tissue had twice as much of the average caterpillar weight as those that fed upon the tomato leaf tissue. Caterpillars that fed on the leaves had higher levels of digestive-related gene expression than caterpillars that fed on the tomato fruits. This included a variety of digestive genes such as proteases, lipases, and carbohydrates. And this table was donated by Diaz. However, you can clearly see that despite the variation in all of the genes, for example, these are proteases, this is a protease and this is a lipase, and there are carbohydrates as well. In every case, the gene expression for the caterpillars that fed upon the leaf tissue is always higher in every single, for all of these. And in some cases, it's basically high to such a degree that it's actually quite astounding. 82-fold difference is nothing to laugh at. This suggested a higher compensatory physiological cost for the caterpillars that fed on the tomato leaves over the tomato fruit tissue. And the energetic cost of extra digestive enzymes can result in a decrease in caterpillar growth. The objective of my project was to determine the differences in anti-nutritive related defense gene expression in tomato leaves in comparison to tomato fruits. This may help to explain the differences in growth and gene expression for caterpillars that fed on the tomato leaves in comparison to those that fed on the tomato fruits, as was shown earlier. The main hypothesis is that there will be higher levels of anti-nutritive plant defense-related gene expression in tomato leaves compared to the tomato fruit tissue. In May of 2017, we planted 20 tomato seedlings in Dr. Musser's garden in Macomb, Illinois. In mid-August, we randomly harvested the plants for around 20 samples of the tomato leaves and fully mature grain fruits. Then the tissues were frozen in liquid nitrogen and stored in a negative 80 degrees Celsius freezer. Afterwards, the leaves and fruits were ground up individually with a mortar and pestle into a fine powder. Total RNA was purified individually from the leaf and the fruit powder using the trisal method following the manufacturer's protocol, in a manner similarly developed by previous researchers in the past. What we essentially did was render the RNA into the aqueous phase and remove the supernatant before washing off the solid sections of the RNA with the ethanol, and then putting it into frozen storage. Afterwards, the total RNA was made into cDNA with the Agilent Low ROX kit following the manufacturer's protocol, which, in other words, we basically just took the total RNA and used this to pull off reverse transcriptase in order to make the cDNA. There was at least 5 biological replications per treatment, 5 for the tomato leaf tissues and 5 for the tomato fruit tissues. And a one-tailed t-test was used to determine differences in gene expression between the 2 treatments. The qPCR was done with the fast CyberGreen master mix and optical 96 well plates in the Step 1 Plus real-time PCR system instrument detailed here, and what this basically means is that we basically just denatured the cDNA and basically use the cyber grains attached to the primers to basically data the specific sequences that we wanted to see, in terms of which is basically referred to as amplification. However, we pretty much had, on another note, a wide array of plant genes were selected, specifically the housekeeping gene of actin, mostly because actin is in every eukaryotic organism, and the anti-nutritive plant defense genes consisted of protease inhibitors, right here, and protein/amino acid limiting genes like threonine deaminase and polyphenol oxidase. Our plant defense signaling genes were cysteamine, alanine oxide cyclase, and S-adenosyl carboxyl transferase. And we use these pathogen defense related genes as well in order to and finally, we basically use the stress-related gene of abscisic acid inducible dehydrin and the photosynthesis gene of rubisco due to the fact that we had to make sure that the leaves were not tampered with, and due to the fact that naturally you can expect the leaf tissue to have more expression of a photosynthesis gene in comparison to the fruit tissue, since that's one of the main functions of the leaf tissue. As for the results, there was a modest trend in increased gene expression of jasmonate signaling related genes such as cysteamine and alanine oxide cyclase. In addition, the plant pathogen related genes such as salicylic acid carboxyl methyltransferase and glutathione transferases all had a trend of being higher in terms of gene expression in the tomato leaves in comparison to the tomato fruits. Now, the interesting thing that you can actually figure out from this graph is that this gene here isn't exactly an anti-nutritive plant defense gene. Instead, it's actually part of another group of genes instead, and yet it has the highest gene expression out of all of the genes in the tomato leaf tissue, which is actually kind of unexpected. However, the plant's jasmonate-inducible anti-nutritive defense-related genes were significantly higher in gene expression in the tomato leaves than they were in the tomato fruits, such as wound-induced protease inhibitors 1 and 2, threonine deaminase, and polyphenol oxidase. There was a bigger increase in photosynthesis-related gene expression in rubisco for the tomato leaves in comparison to the fruits, but that was to be expected in the first place. And strangely enough, these two genes are closely related, and yet there's quite a bit of variation between the two in terms of gene expression for the tomato leaf tissue. That was pretty unexpected. However, one thing we did expect was threonine deaminase being the having the highest gene expression in the tomato leaf tissue out of all the genes tested, due to the fact that this was a recurring trend in previous research done by others in the past. What wasn't expected was the large degree of difference between the tomato leaf tissue and the tomato fruit tissue in terms of gene expression. After all, we have differences that range from around a 500-fold difference to even a 1,000-fold difference between the two tissue types, which is honestly quite unexpected. The plant pathogen-related genes such as acid phosphatase, pathogenesis-related protein precursor 1A1, and acidic endochitinase were all higher in the tomato leaf tissue than in terms of gene expression than in the tomato fruit tissue. There was a higher increase in gene expression for abscisic acid-inducible dehydrin, which is a drought stress-related gene for leaves in comparison to the fruits. As can be noticed here. However, once again, the genes that were basically the highest in terms of gene expression weren't the anti-nutritive plant defense-related genes. However, this graph actually came from the results of a different plate. Also, another thing to note is that you'd think that the genes that were influenced by the salicylic acid pathway would actually be much less regulated in terms of wouldn't be as highly regulated in terms of gene expression in the tomato leaves due to the crosstalk between the two pathways. Yet in this case, there's still some upregulation anyway. What this ultimately says is that maybe there's a small chance that perhaps the tomato leaf tissue is much better at basically defending against pathogenic infection and herbivory in general in comparison to the tomato fruit tissue, but further research needs to be conducted in order to confirm this. Our results support my hypothesis that the tomato leaves have higher levels of gene expression compared to the tomato fruit tissues. The higher levels of anti-nutritive plant defenses in the tomato leaves in comparison to the tomato fruits might explain in part why the caterpillars that feed on the tomato leaves grow slower and stimulate more digestive-related genes than caterpillars that feed on the tomato fruit tissue. However, as stated earlier, there may be other explanations, such as the levels of available nutrients in the two types of tissues being a possible reason for the difference as well. Our project helps develop a framework on caterpillar selection of plant tissues. I'd like to basically thank my advisor, Dr. Musser, for helping me out through this entire ordeal, and I'd like to thank Dr. Musser for basically being as helpful as she was, and I'd like to thank Dr. Kenneth Gravy for